This video is dedicated to the most common instrumentation that you can use to work with biological molecules, for example, proteins, when using mass spectrometry. All mass spectrometers work in the same principle. There is an area in which the sample is introduced and they have what is called an ionization source, a way of making that sample into gas form and ionize. Then a way of separating the different ions form according to their mass to charge ratio, and then a detector, a way of detecting that signal and those different mass to charge ratios and the relative abundance of the different ions that produce those mass to charge ratios. So this is how all mass spectrometers look like. In protein identification or in biological applications of mass spec, the ionization source are either MALDI or electrospray. The ion separations can be time of flight, quadrupole, or ion traps. And finally, as ion detection methods, you can use a photomultiplier, a microchannel plate, an electron, or an electromultiplier. And in this video, I will be looking briefly at the ion sources and the ion separations that make the most common mass spectrometers used for mass spec of biomolecules. Why is instrumentation important? Well, it makes a huge difference in the amount of information you can get from a mass spectrum and whether you can identify a biomolecule on the basis of its mass spectrum. The key parameters are the accuracy of the mass, the resolution of the spectrum, and the sensitivity of the equipment. And it makes a huge difference. This is, for example, a molecule of chlorophyll A, and this is one mass spectrum that shows the peak of the molecular mass of chlorophyll A. Chlorophyll A has an average mass of 893.5. However, the monoisotopic molecular mass, which would be the mass of the compound but with the most common isotopes, carbon 12, oxygen 16, nitrogen 14, is actually 892.5. With a better mass spectrometer, you can start getting a resolution of peaks. This is not only one big peak, it's actually a mixture of the different peaks produced by the different chlorophyll A that you have in the sample, depending on the isotopes that are making that chlorophyll A. And if you have an even better mass spectrometer, then you can really have a very, very good resolution of peaks. So you can identify the different isotopes that make those peaks, and the intensity of the peaks tells you information about how abundant that particular ion is in your sample. So as you can see, with the same compound, you can get much more information if you have an accurate and a mass spectrometer with high resolution and high sensitivity. So a lot of work has gone in the last 2 or 3 decades to improve the instrumentation to make better mass spectrometers that allows us to identify biomolecules more successfully. A little bit of going back to basics to remind us what all this is about. The first step of any recording any mass spectrum is to ionize the sample in a gas form. So you produce ions of your compound in gas phase by electron ionization. Once you have that ionized molecule, what we call the molecular ion, then there's also a fragmentation. It is not only preserved as one ion, it fragments through the weakest bonds and to produce a series of ions, and they can be even ions or they can be odd ions, and all those ions are then recorded by the mass spectrometer. once the ions are produced, they are separated in the mass spectrometer according to their mass to charge ratio, and they are detected in a proportion to their abundance. So you will get higher peaks from the most abundant ions. And that is what the analyzer and the detector parts of your mass spectrometer do. And that's when you finally have a mass spectrum of that particular molecule of interest with a peak that corresponds to the molecular mass, but also assorted other peaks that correspond to the fragmentations, the different fragments that molecule has broken down into in your mass spectrometer. So if we look at a very simple molecule, for example, the molecule of methanol, this is the mass spectrum of methanol, and you can see that you have the peak that corresponds to the molecular ion, which is 32, the molecular mass of the compound, but you also have the most intense peak, the most abundant ion, which may or may not be the molecular ion. Very often it is not. In this case, is the peak at 31. That most intense peak is called the base peak, and it's given it's giving a value of 100%. And all the other peaks are plotted relative to that peak. And that is when why the y-axis of our mass spectrum is called relative abundance. How abundant are all those peaks with respect to the largest peak that is given the value 100. Do you get an average molecular mass for your compound, or do you get monoisotopic masses with the most abundant isotopes and subsequent combinations? Well, it depends on the sensitivity of the instrument. If it's good enough, you do not get the average mass, you get monoisotopic masses. For example, this is a mass spectrum of a peptide fragment, 8, 10 amino acids, and the average mass of this fragment would be 1086.28, but this spectrometer is sensitive enough and has good enough resolution that it's giving you the different masses or mass to charge ratios for the different isotopic ions, including the monoisotopic mass, the mass of the compound with the most abundant isotopes. Ion sources. MALDI-TOF is one of the very popular ones. MALDI stands for matrix-assisted laser desorption ionization. And the ions are produced, the sample is introduced in the equipment, mixed with a matrix. UV laser light pulse is used to produce the ions. And here's just a sort of a schematic of how the MALDI ionization source works. Your molecule, your analyte, your molecule of interest is mixed with a matrix mixture and injected into the spectrometer. It's a laser beam is shone through, and it breaks down the matrix analyte mixture, and different ions are created, and those are the ones that go through to the next step, to the analyzer. The other main ion source for mass spectrometry of biomolecules is electrospray. And in an electrospray, the sample is injected through an electrospray needle. It becomes a gas, it goes through some charged electrodes, and that creates the ions. And then once the ions are created, again go through a separation process and then are detected. So basically and you have a picture here in which you can just about see the needle spraying the sample very fine, and it's with electrodes the ions are produced. An interesting challenge of using ESI to produce you as an ion source is the fact that for one particular mass, for one with a particular isotopic set of isotopes, it can produce more than one peak, and that is because it can produce more than one charge ion for that particular fragment, and that means different charge to mass ratios. So instead of having one peak for a particular fragment, you have a series of peaks, and that's what I'm showing in this slide. It's called the mass charge envelope. What you're aiming for is one peak, a peak that corresponds to that particular fragment, and you can do that by using something called the convolution. So it's just an algorithm that transforms a spectrum that contains multiple peaks for a particular mass, for a particular charge species, for a particular mass of a particular fragment, because you are generating in the ESI process several charges and several mass to charge ratios into one. Equipment, commercial mass spectrometers have the appropriate software, and they will already apply the convolution algorithms to the so you get the convoluted spectrum, one peak for one particular fragment.